Hello, so another video from me. We're going right back to the start of the year now with cells and organelles. So there are three kinds of cells you need to know about: eukaryotic animal cells, eukaryotic plant cells, and prokaryotic cells, which are like sort of bacteria and archaea. So let's have a look at the eukaryotic animal cells. They're about anywhere from 2 to 200 micrometers, mostly around the top end of that scale, so quite large as cells go. They have many internal organelles, which we'll go through in a minute. They have a sort of very unfixed shape, and in the human body there are many different highly specialized types of animal cell, which all derive from stem cells. Plant cells are pretty much the same, except they have a few extra features. They're about the same size, but they also have chloroplasts, a large central vacuole, and we call that a large central vacuole because sometimes in animal cells you can get small vacuoles like phagosomes. So we call this one a large central vacuole just to distinguish it, and also a cell wall around the outside, which between them provide external and internal support for the plant cell, because remember, plants don't have skeletons. So they have those three extra organelles, but they also don't have any centrioles, which is a bit confusing because centrioles are really important in cell division, and no one's quite sure how they control their cell division without centrioles, so that's quite interesting. So it's quite rigid, and it's filled with water to help keep the whole plant turgid. That's the word we use to describe when they're sort of full of water and supporting their cells, supporting their stem, they are turgid. The opposite of this, when they haven't got enough water, is flaccid. I know, hilarious. Okay, so about the same size as these small organelles like mitochondria and chloroplasts are the prokaryotic cells, which are usually less than 2 micrometers. Very, very small, almost like a nothing, and they're about 2 micrometers. They've got no membrane-bound organelles. To replace those, they have this little infolding called a mesosome, which is where all the reactions that would normally happen in smooth and rough endoplasmic reticulum, chloroplasts, mitochondria, all those reactions just take place on this one place, the mesosome. Got no nucleus, no other organelles apart from ribosomes. That's a big loop of DNA, and they also have these smaller loops of DNA called plasmids. They do, however, have a cell membrane, a cell wall, which is made out of peptidoglycan or murin, and also sometimes they have this like slimy outer layer, slime capsule, which helps protect them from the world they're living in. They can also have things like flagella and pili to help them move around, and cilia to help them get towards their food sources. Okay, one little thing about their ribosomes is the ribosomes are a bit smaller than the eukaryotic ones. So eukaryotic ones are called about 80S, or about 30 nanometers, but in a prokaryote they're about 70S or 20 nanometers. They're a bit smaller. Okay, so they do have ribosomes, but they're kind of different. Right, let's have a look at all of the eukaryotic organelles we need to know about. So let's start over here with the nucleus. The nucleus is sort of the brain of the cell. It has a nuclear envelope around it, which is a double layer of membrane with holes in called nuclear pores. These are just 2D drawings. Sorry about my lack of 3D drawing skills. The middle of the nucleus is filled with DNA, and when it's unwound, we call that chromatin. It's kind of like a mesh of DNA. And there's a particular dense part of the chromatin called the nucleolus, and that's where ribosomes are made. Okay, so things can get in and out of the nucleus through the nuclear pore. You can get transcription factors coming in, you get mRNA coming out, but it's also there to help protect it from larger molecules getting in and damaging the DNA. So it's all about keeping that DNA safe and sound. So the DNA doesn't leave, but mRNA can. Next is a mitochondria. So these are powerhouse of the cell in plants and animals. Mitochondria, I could do a whole other video on them. They're really quite complicated. It's where aerobic respiration happens. You cannot just say respiration. To get marks, you have to say aerobic respiration that uses oxygen. It's got an outer and inner membrane, which has an intermembrane space between them. The inner membrane are folded into cristae. They're so complicated. They have their own ribosomes inside, and the inside's called the matrix, because it is like another world, I suppose. And they also have a little loop of maternal DNA, which means it perfectly matches your mother's, instead of the DNA in your nucleus, which is half mum, half dad. Chloroplasts are quite similar in size. Remember, these are about the size of a prokaryotic cell, so they're quite small, about 2 micrometers. The chloroplast also has an inner and outer membrane with an intermembrane space, and it has these stacks of thylakoids called grana. The thylakoid membranes, that's where photosynthesis actually happens. They've also got their own ribosomes and DNA, but the middle part is called stroma instead of matrix. So sticking with plants, that vacuole we mentioned earlier has a special membrane around it called the tonoplast, and the cell wall surrounding the plant cells is outside the cell membrane, and it has this really complicated mesh structure of cellulose, which is held together with hydrogen bonds, and it's totally permeable to water, so it doesn't really stop anything from getting to the cell, it's just there for support. And remember, it's 3D, so it goes all the way around. It's not just in my 2D diagram going around the outside, it's all the way around, so it really supports the cell. Right, next are ribosomes, which for the most part on any diagram or electron micrograph you'll see are like a dot. But when you look closer, they have a small subunit and a large subunit stuck together, which looks a bit like a mini snowman. So these make proteins. They stick together amino acids to make complex polypeptide proteins. And sometimes those ribosomes are found on endoplasmic reticulum, so I'll come back to that. Vesicles are little membrane sacs that transport things out of the cell, or into the cell, or just around the cell. They usually use the cytoskeleton to get around by sort of walking along it using dimer proteins. If they fuse with the external cell membrane, which is what's going on here, and release their contents into the outside world, it's called exocytosis. And if things are coming into the cell the other way, it is endocytosis. Very, very simple. Remember, membranes are just like squishy nothings. They just go around joining onto stuff. It's all very fluid. That's why it's called a plasma fluid membrane. Okay, so back to the endoplasmic reticulum. This is an extension of the nuclear membrane. So this is the nuclear envelope here, and then it extends off into these flattened sacs called cisternae. If there's no ribosomes, then it's smooth endoplasmic reticulum, or SCR for short, and that synthesizes and processes lipids, or fats, to you and me. And the rough endoplasmic reticulum does have ribosomes, rough endoplasmic reticulum, RER for short, and this synthesizes and processes proteins. So ribosomes, if they're just in the cytoplasm, the inside of the cell, they make proteins that are going to stay inside the cell. But if you want to make a protein that's going to leave the cell, like, I don't know, keratin, that's going to go out to make your nails, then you need to be able to put it into a vesicle to leave the cell. So if you want the protein to leave the cell, it has to be made in the rough endoplasmic reticulum, so it can be pinched off into a vesicle from the edge of the rough endoplasmic reticulum, like here, you know, pinched off the edge, and leave the cell by exocytosis. Next we've got cilia and microvilli, which are both extensions in the cell plasma membrane. Microvilli, you find them in places like the kidneys and the lining of your gut. They increase surface area for absorption, so they're just extensions in the cell membrane, really. They're just, you know, in out, shake it all about. And the cilia are the exact same, except they've got microtubules inside, which allow them to move. So you find these in places like your trachea, so they can move dust out of your lungs, so they actually move back and forth. They can wave like a Mexican wave. Cell surface membrane, probably going to make a whole other video about these, but it's basically a phospholipid bilayer once you zoom in, but mostly you'll just see it as a black line surrounding the cell underneath the cell wall in plants. Lysosomes are like vesicles, but they're full of hydrolytic enzymes which help to recycle old cells. Golgi apparatus help to process and package things that have been made by the smooth endoplasmic reticulum, and they also make lysosomes. Stuff comes in at the cis side and out at the trans side. And finally, we've got a few extra things involving the cytoskeleton. The cytoskeleton is a series of tubules and meshed microtubules that help support the cell and move it around. So amoeba use their cytoskeleton to move around. The centrioles make them, especially the microtubules. They're also involved in cell division, like mitosis and meiosis. And a flagellum is another word for sort of the tail of cells. So the only ones that you find in eukaryotic cells are sort of like sperm cells and other gametes sometimes have flagellum, which also have microtubules inside, a bit like the cilia, that help them to move around and swim around. Okay, so that was a very quick summary of all the organelles. Thanks for watching.